would like to thank the organizers for inviting me to share some of our work. And we also, like the previous speakers, are funded with an Excellence in Hemoglobinopathies Research Award. And our award is focused around studying the cardiac, pulmonary, and renal complications of sickle cell anemia. So as this is not new to this audience, that vaso-occlusions are the hallmark of sickle cell disease. But their absence does not prevent multi-organ damage, and multi-organ damage can occur without vaso-occlusions, which was us thinking outside the box, because the current paradigm or dogma is that all the organ damage, all the consequences of sickle cell disease are actually mediated by sickling. We have actually data to show that it's the sickling related signaling that can damage the organs over time, and it doesn't have to directly cause sickling. And so we've been studying the mechanisms for chronic organ damage with this U01 award. And we've been studying, for instance, it's assumed that renal pathology has classically been assumed to occur due to sickling-induced ischemia and necrosis. And therefore, I think a lot of the molecular mechanisms that underlie the chronic organ damage have not been explored. So my talk today will focus a little bit, I'll just touch upon sickle nephropathy and then describe the sickle cardiopulmonary complications we've been studying in a bit detail. And I apologize to people who heard the cardiac talk yesterday at the cardiopulmonary symposium. There might be a little bit of overlap there. So this is just to give you an idea about how the kidney structure is for those who are not as familiar with this. So I classify the sickle nephropathy in two parts. It's a glomerulopathy, which affects the glomerular or the cortical area, and a tubulopathy, which is where the loop of Henle, this is the glomerulus, and this is the loop of Henle surrounded by the vasa recta, that creates the osmotic gradient, allowing urine concentration. And this portion, which resides in the medulla of the kidney, is actually indeed prone to sickling, because the medulla is a very hypoxic, hyperosmolar, acidic environment. All these factors promote sickling, and therefore there is sickling of red blood cells in the vasa recta, and this removes the osmotic gradient. Because of the sludging of blood flow, the counter current is lost, and urine concentration defect develops very early, and this indeed is as a result, direct result of sickle hemoglobin. In fact, there was a very nice paper by Marty Steinberg in 1971 where he showed that sickle trait patients also will develop a urine concentration defect in the 3rd to 5th decade of life. And he found that it depends on the number of alpha genes. So if you had only 2 alpha genes or 3 alpha genes, you have proportionately less sickle hemoglobin compared to patients with 4 alpha genes. And the amount of urine, the onset of urine concentration defect in sickle trait patients was directly dependent on the sickle cell gene dosage. So I won't be focusing on this part because this is known and very well studied. The thing that has perplexed us is the glomerular disease, which is actually the cause of mortality, because patients with sickle cell disease have a urine concentration defect that begins as early as infancy and lasts throughout their lifetime. But what kills them is development of glomerular disease leading to proteinuria and focal segmental glomerular sclerosis, followed by end-stage renal disease. So I'll skip this because I think I'm already out of time as I start. So sickle glomerulopathy, I just alluded to the details of this. So what we did is our lab discovered that in sickle cell anemia, Sickle cell anemia per se results in hyperangiotensinemia, which occurs in the absence of hypertension. So this is the plasma angiotensin levels in wild-type mice versus the humanized sickle cell disease mouse model, the Berkeley sickle mouse you've heard Solomon and Kalpana talk about. So very significantly high levels of angiotensin. Yet patients and mice with sickle cell disease have blood pressures that are actually on the lower quartile of normal because of the hyperdynamic circulation. So angiotensinemia, hyperangiotensinemia does not associate with hypertension in sickle cell disease. And we confirmed this in children with sickle cell disease who were all normotensive. These are all children and their age, somewhat age-matched sibling controls, unaffected siblings. And we found plasma angiotensin 2 to be high. So based, and I'm not going to belabor on all the mouse data, we found that this actually directly results in the FSGS, end-stage renal disease, and glomerulopathy that I talked about. So based on this, we conducted a multi-center phase 2 trial of blocking the angiotensin receptor in sickle cell disease. This trial was conducted by Charles Quinn as the principal investigator. It was multi-center. And some of the investigators who were part of this trial are listed here, and some of them are here at this meeting, and I was the sponsor investigator of this trial. And basically we enrolled patients that had no albuminuria, microalbuminuria defined as urine albumin of 30 to 300, and macroalbuminuria. And they received 6 months of losartan therapy. And then they were assessed before and during the treatment and after the treatment. And again, due to the shortage of time, I'm only going to present the key data. So we found the percentage of patients, so our endpoint was that we have more than 25% reduction in urine albumin creatinine ratio from baseline in a third of the patients. And we actually, with albuminuria, and we, the study met its endpoint. We had a 58% reduction in the microalbuminuria group. And actually, we were very surprised. We had powered our study on the microalbuminuria group because we had assumed that patients with gross albuminuria or macroalbuminuria, the horse is already out of the barn, and we probably cannot reverse the nephropathy. We actually had the most effective response in the macroalbuminuria group, and this manuscript is actually submitted for, is under review right now, which will have all the detailed data, and I just don't have the time to show it in a 20-minute talk. So we are actually, this trial was designed as a pilot and feasibility study to see how many patients we need to power a phase 3 randomized placebo-controlled trial, which is currently being designed and will open at all the sites who participated in the original trial. And we also will enroll a few other sites, including our long-standing collaboration now with the Caribbean Institute of Health Research in Jamaica. So now I'll switch gears to cardiac pathology in sickle cell disease. All of you are probably very familiar that patients with sickle cell disease have increased stroke volume and increased cardiac output, all secondary to the anemia and the hyperdynamic circulation, which leads to chamber dilatation. And this chamber dilatation is usually pan-chamber dilatation. They have normal to hyperdynamic systolic function. And these are the 3 features that have actually prevented any further look into the cardiac pathology, because all of the cardiac features have been assumed to be due to the anemia-related hyperdynamic circulation. But more recently, studies have shown that patients with sickle cell disease actually develop diastolic dysfunction. Atherosclerosis is uncommon in sickle cell disease, and myocardial hemosiderosis is also very uncommon in sickle cell disease. The mortality in sickle cell disease in the resource-rich countries still is much lower than that seen in normal people in those countries. And the cause of death, one of the causes of death in sickle cell disease that was shown even in the comprehensive sickle cell disease study done decades ago, was sudden death in about 30 to 40% of patients. And autopsy studies have shown that there is sudden unexplained death in about 20 to 40% of patients with sickle cell disease. And the causes of death currently, at least in the US, are cardiopulmonary, and renal actually takes the rest of it. And this was a study done by Mark Gladwin in 2004. This is a landmark study where he showed that patients who had very mildly elevated pulmonary arterial systolic pressures that were measured by a surrogate marker called TR jet velocity, and if TR jet velocity was greater than 2.5 versus less than 2.5, there was a very significant increase in mortality. In fact, the odds ratio, they were 10 times higher risk of death than patients with a TR jet velocity less than 2.5 meters per second. This is just the same data showing what was the proportion of this is normal, and these were the sickle patients he showed. These are 2.5 to 3, and these were patients with TR velocity over 3 meters per second. And this actually relates to, this can be extrapolated to a pulmonary systolic pressure of 25, 35, and 65. And some of you are familiar with primary pulmonary hypertension know that the pressures in primary or idiopathic pulmonary hypertension, the pulmonary pressures almost reach systemic blood pressures, and these are very mild elevations in the systolic pressure. It's elevated TRV over 2.5 will be the criteria, that is the criteria used for in sickle cell disease for elevated TRV, is seen in about 30% patients with sickle cell disease. And I've already said that this is very modest, and it has been shown that the elevated TRV, which was originally thought to be synonymous with pulmonary arterial hypertension, is indeed not the case in sickle cell disease. When right heart cardiac catheterization was done, only about 25% to a third of patients who have elevated TRV actually have pulmonary hypertension. Nevertheless, elevated TRV is a very significant marker for mortality in sickle cell disease. So this is just the summary of what I just spoke. What is known: high prevalence of increased TRV. The pulmonary arterial hypertension is actually less frequent than the pulmonary venous hypertension, present in about a third of patients with elevated TRV. And there is LV dilatation, hypertrophy, diastolic dysfunction, and a high rate of sudden death. So this is the state of the field in cardiac pathology in sickle cell disease. And we were looking for if there is a unifying pathophysiology that can explain these findings, and what are the causes of cardiac death in patients with high TRV without pulmonary hypertension. So we first performed a prospective study of all analyzing, it was a combination of prospective and retrospective study where we analyzed echocardiograms of 134 children at our hospital with a mean age of 12.3 years. So these patients are really not at a stage where they have extensive organ damage and extensive cardiac pathology. They don't even have pan-chamber enlargement at this age. And one of the features that struck us was that and all of these echoes were obtained at baseline or steady state. We found that left atrium, they all had dilated left atria, and that was that was highly significant in this study. We found that these patients had a proportion, 8 to 14% of children with sickle cell disease had evidence of diastolic dysfunction by echocardiography, and these are the indices you will see often listed as measures of diastolic dysfunction. And the diastolic dysfunction actually increases with increasing age in this cohort. We went back and analyzed data on about 4,000 patients, doing a meta-analysis of all the cardiac imaging studies reported in sickle cell disease. And we showed that left atrial diameter, if this is normal, in almost all the studies, whether pediatric or adult, there was increased left atrial diameter, which was increased in proportion to the other chamber enlargements. We also found that 24 of 26 studies reported evidence of diastolic dysfunction. Prevalence was 17 to 77%, and severe diastolic dysfunction was present in about 1/4 to 1/3 of patients. When we looked at TR jet velocity reported in these studies, we actually found that the weighted mean average of TR jet velocity was 2.34, and some studies there was increased TR jet velocity, but in a lot of studies the TRV was not elevated, and this and pulmonary hypertension in those studies with right heart cath showed that, as I said before, pulmonary hypertension was present in about 1/3 of those patients with elevated TRV, of which the majority of it was pulmonary venous hypertension. So based on this data, we decided that we need to do a longitudinal study, because the data that is present is mostly inferred from cross-sectional analysis. There are limited right heart cath studies in patients of different ages, and there's almost no left heart cath studies to study the diastolic dysfunction, which primarily is a left ventricular dysfunction. And the bulk of the cardiac data reported in sickle cell anemia is based on echocardiography and with very limited cardiac MRI studies. So we designed a study in sickle cell mice where we could study this longitudinally in a relatively short time. And these mice underwent the exact same imaging as humans with sickle cell disease. We had cardiac echoes, MRI, EKGs, and in addition, we have a distinct advantage of obtaining cardiac histopathology and the molecular analysis with time. We first proved that the sickle cell mouse model was representative for humans with sickle cell disease, and indeed it was. There was evidence of hyperdynamic circulation, anemia-related increased left ventricular mass, normal ejection fraction. So this essentially shows a hyperdynamic physiology with normal systolic function. I won't belabor on the individual parameters, besides I'm not a cardiologist anyway. So, and then like humans with sickle cell anemia mice also have evidence of diastolic dysfunction. So the sickle cell mice are denoted by the filled triangles and the wild type mice by the open triangles, and the relaxation rate and the E by E prime ratios were significantly higher. Then we followed these mice with serial echocardiograms, and we see that if mice were 1 month of age, which is soon after weaning, the sickle mice are a little smaller in size because of the anemia, and their left atria were actually a little smaller, though not statistically significant, than wild type mice at 1 month of age. But as the mice grew in age, there was left atrial dilatation that was progressively increasing, while there was no such thing seen in the wild type mice. And this happened in the absence of any mitral regurgitation, which can of course cause left atrial enlargement. To prove that whether this was a consequence of the anemia, we actually experimentally induced anemia in wild type mice to the same degree and duration as the sickle mice. This was specifically related to sickle cell disease. And this is just a section of the hearts. This is the 8-month-old wild type mouse, iron deficiency anemia mouse, 3-month-old sickle cell anemia mouse, and a 7-month-old sickle cell anemia mouse. We then did analysis of, we hypothesized that this is probably due to diffuse myocardial fibrosis causing diastolic dysfunction, and therefore the left atrium is not able to empty itself into the left ventricle. So we measured something called extracellular volume, a measure of diffuse myocardial fibrosis, and indeed this was significantly elevated in mice with sickle cell disease. And if we transplant sickle hematopoiesis into normal mice, we see the ECV gets elevated, showing that it's a direct consequence of sickle cell disease. And we proved that by histopathology. We see evidence of diffuse myocardial fibrosis. And based on this, we designed a clinical study to see if this is indeed the case in humans with sickle cell disease. I think I'm running out of time, so I'm going to rush through this study. We basically took 25 patients, children, young adults and adolescents and adults. These groups were with or without elevation of TRV, and we studied these, and this slide summarizes the data from the entire study. So every single patient with sickle cell disease had ECVs that were higher than normal. These two lines denote normal. These are normal age-matched controls, and ECVs were elevated in all the patients, and there was no evidence of iron overload. And when we compared the degree of myocardial fibrosis seen in other cardiac diseases, sickle cell disease was the highest, and it was only less than the ECV of scar tissue from a myocardial infarction. So this was a very significant finding, and we then when we analyze our data in detail, we find if we take the patients with elevated ECV and divide them arbitrarily as relatively low fibrosis versus high fibrosis, we find that patients with normal diastolic function in this cohort have less ECV elevations than those with diastolic dysfunction, suggesting that cardiac fibrosis predates diastolic dysfunction. And another thing that was very significant was that patients with diastolic dysfunction had significantly higher anemia. When we look at TRV and the red circles here, if I want you to focus on this last lower F, panel F, which has kind of masked this restrictive physiology, impaired diastolic filling, increased left atrial size, and modest pulmonary venous hypertension that develops. So, and a huge group of people, it's almost a village that has taken a multi-divisional effort to get these studies done. And with that, I will end my talk. Thank you.